Now, a very important mechanism in innate immunity is phagocytosis. This means cell eating. Now, there's certain cells which are phagocytic. They are capable of eating bacteria. And in fact, phagocytic cells can also eat dead tissue cells, which is good. Because if you have a wound, you don't want lots of dead tissue cells hanging around because bacteria can infect those, leading to infection. And the phagocytes are actually types of white blood cells. Neutrophils are the most abundant type. And also there's monocytes, and when monocytes are in the tissues, they become macrophages. So circulating in the blood you have neutrophils and monocytes, and if there's an inflamed or an infected area, neutrophils will migrate into that area to eat up the bacteria that are causing the infection. After that, it takes a bit longer, but it will still happen, monocytes will migrate into that area, and the monocytes in the blood will start eating bacteria, and they become really big, huge cells called macrophages. So the neutrophils will migrate from the blood and into the tissues and start eating, and the monocytes will, but when the monocytes move into the tissues, they change their name and become macrophages. But in actual fact, even in tissues all the time, there are some macrophages hanging around just in case infection does become a problem. So there's some tissue-based macrophages, but they can be reinforced by monocytes from the blood. And they take part in this process called phagocytosis, or cell eating. So let's think about what happens. Well, here's a couple of bacteria which have got into the body somewhere, perhaps from a wound. And the chemicals given off by these bacteria are recognised by the macrophages. And the macrophage, let's have this is a macrophage. It could be a neutrophil, could be a monocyte, but this is a macrophage. Nice big cell. That will move towards the infecting bacteria. So that's the first thing. It will recognise the bacteria and move towards it with chemical attractions. It's chemically attracted to the bacteria. So you end up with a situation now where you have the bacterial infecting organisms here, and the macrophage is now right next to it like this. So that's the next stage in the process. Now what happens now is the macrophage keeps on moving and it starts to engulf the bacteria. So the next stage is, here's the bacteria. The monocyte keeps moving and it starts to engulf the bacteria like this. So it just keeps moving and starts to engulf the bacteria. And then what happens is that these two arms of the macrophage, they can actually join together and they will fuse. So if we look at this a little bit of time later, here's the bacteria as before. But now the arms of the monocyte have virtually completely surrounded it because it's kept moving. Now the cell membranes in macrophages, like all cells, are made out of lipids. They're phospholipid layers, and because they're lipids they can just flow together and merge. So what happens is these simply merge, these membranes merge. So the situation now is the bacteria have been completely engulfed in a membrane-bound area inside the cell called a phagosome. It's now engulfed inside the cell. So the situation now is that we have the bacteria in this phagosome inside the cell. There's the cell. So they've been eaten. But the trouble is the bacteria are still alive inside there. They're inside the cell, but they're still alive. So what we need to do is kill them off. So fortunately, inside many cells, particularly inside these eating cells, inside phagocytes, there are small areas, small vesicles throughout the cell, and these small vesicles are called lysosomes, and the lysosomes are little packages of digestive enzymes, one of which is called lysozyme. So the lysosomes contain lysozyme. And here we have a phagosome, and what happens now is that the lysosomes containing the lysozyme move towards the phagosome containing the bacteria. That moves towards it. So now we have a situation where we have the cell membrane of the phagosome and the cell membrane of the lysosome next to each other. And because they're fluid membranes, they will flow into each other. So the same thing again happens. These membranes flow into each other. So we have a situation here where the membranes join together, like this. So what this means is there's now a complete gap between the membranes. So let's draw that in bigger. Here's the phagosome, and here's the lysosome, which is now merged with the membrane. Here's the bacteria inside the phagosome. But now, because the lysosome has merged with this phagosome, it means the digestive enzymes and the lysozyme can now get in to the phagosome and will digest the bacteria, will digest and kill the bacteria. This is good because it kills the bacteria. It breaks them down into simple compounds like amino acids, glucose, fatty acids, and these are actually simply absorbed into the cell, and the cell uses them as nutrients to grow. Now when this process is finished, there are actually maybe a few little cell fragments left that are indigestible. So what happens is that the phagosome, which is now combined with the lysosomes, moves towards the outer cell membrane, the membranes fuse, and the indigestible products can be ejected from the cell. So really good mechanism, phagocytosis. Bacteria and dead tissue cells are ingested, digested, the bacteria are killed, the nutrients are absorbed. And this works particularly well because the immune system labels bacteria which need to be destroyed. Now, when there's infection in the blood, the presence of that infection will act on certain proteins in the blood called complement proteins. These complement proteins complement the activity of the immune system. So bacteria get into the blood. Proteins in the blood are triggered into becoming other types of protein, and this new sort of protein has immunological functions. It's called complement. This is part of what's called the complement cascade. So what this means is the presence of infection in the body will stimulate the triggering of complement, and that means that new proteins which weren't in the blood before will become present in the blood. And some of these proteins will stick to infecting organisms. And we see this in this diagram here. Here we have the pathogenic microorganism which is causing the infection. And it's got specific recognisable antigenic molecules on the outside of this. And here we have the phagocyte waiting to do the phagocytosis, the ingestion and the digestion. And these activated complement molecules, the activated protein in the blood, form opsonins. And the opsonin will stick onto the bacteria, stick onto the antigen. And the presence of these opsonins on the surface of the bacteria allow the phagocyte also to stick onto it. They're like a bridge, if you like, gluing the bacteria to the phagocyte. It glues them together. They've been opsonized. And that means it's very much easier for the phagocyte to keep hold of the bacteria where it can ingest it. The opsonins and the receptors, of course, in real life would be quite a bit smaller than this. So that's the process of phagocytosis, an essential mechanism of immunity to ingest and digest bacterial cells and viral cells, viral particles.